It is not necessary that always we should transfer these particular genes from plant. So these are also transferred from bacteria or insect or other unrelated plants in which taxonomically cross is not possible. So where that particular genetically modified plants are there. So these are the advanced breeding lines. So there is a question. So genetic erosion is there. So why that particular genetic erosion is there? Because of releasing of popular high-yielding cultivars. So everyone want to take that particular high-yielding plant in his or her field, and because of which whatever such kind of land races are there, or wild relatives, or especially land races and modern cultivars. So these are day by day showing that decrease in area under cultivation, and because of that we observed that genetic erosion. Genetic erosion means loss of that particular diversity of crop plant from the field or from cultivation. And there are certain examples that how that particular improved cultivars or improved lines which are released for cultivation from wheat or rice or other crop, which has applied pressure on survival of these particular land races. And this particular monoculture is very a threat for much or causing a threat as far as loss of that particular plants are concerned. And again, monoculture means cultivation of one type of variety in large area. Say for example, one variety in case of sugarcane, like which is that particular cultivar, 62,000 is there, or 86,032. So these are the particular varieties of sugarcane, or in case of soybean or jawar and so on. So those varieties, if these are cultivated throughout Maharashtra, then what will happen? These varieties will apply pressure on survival of pathogen or insect, and because of which again they become susceptible. So ultimately whatever varieties which we are cultivating in that area, they become after some time they become susceptible. So there should be a diversity in those plants. Now, solution for genetic erosion. So what we have to do, we have to conserve the genetic resources of this. So if we follow this particular practice of conservation of this particular germplasm, then definitely it will be useful. So considering this, in 1972, one conference was held at the UN, United Nations Conference at Stockholm, where it was recommended that there should be conservation of that particular area or that particular plant type which are having diversity. So rich in genetic diversity. So wherever such kind of areas are there, we should either protect that particular area, or we should collect that particular plant material from that area, and we should conserve them, so that not only immediate, but after 50 years or 100 years, that particular plant material can be used by the breeders for improvement. So in that case, two consultative group of international agriculture research was established, that is CGIAR, in 1974, and that particular CGIAR has given guidelines how to conserve that particular germplasm from different sources to one location, and then that particular material can be used for further conservation and improvement of that particular plant. So in that case, need for conservation of plant germplasm. So why we have to conserve that particular germplasm? So in that case, once a particular plant type is lost because of monoculture of improved plant material, so that particular germplasm or land race will lose permanently, and we can't create that particular plant material. So if we conserve that particular material, so in future it may help to provide a solution as far as nutrition value is concerned, or fodder value is concerned, or it is providing a solution against biotic or abiotic stress resistance. So considering that one, or somewhere it is also providing a solution for your medicinal value is there, or it is providing some value against that particular fiber is concerned, and so on. So considering that, we should conserve that particular germplasm. Then if we want to conserve that particular germplasm, then we should understand from where we have to collect that particular material and then we have to conserve it. So for that purpose, we should know what are the particular center of origin of crop plant. So this particular concept of center of origin of crop plant was proposed by Vavilov, and in that case, that particular Vavilov has in this case, we should understand the importance of plant genetic resources. So what are those plant genetic resources? So these are the total sum of all allelic resources or breeding material which can be used for further breeding program. So this particular genetic wealth is acquired by the plant over millions of years. Through the process of evolution, they have acquired this particular diversity. And if you collect that particular plant material and if you preserve it, definitely it will helpful for further conservation as well as improvement of crop plant in future. So in that case, if we see that particular, this picture will tell, so these are the particular wild species, and during the process of domestication, some of the undesired plants or unwanted plants were eliminated, and later modern varieties means that breeder has developed these particular varieties by introducing or by transferring the genes of interest into popular cultivar and they release. So because of, so somewhere at the initial we were here where tremendous diversity was there, then domestication during so as agriculture has started 10,000 years ago to till date, so day by day that particular diversity is decreasing, and again development of modern varieties, again that diversity is declining. So what will happen because of this, if in future if something goes wrong with our cultivated plants because of evolution of new race of pathogen or new race of insect and so on, so these modern varieties will become susceptible, and in that case if you want to improve them, then it becomes difficult because we don't have the germplasm stock. So what we have to do, we have to conserve that particular germplasm, and then we have to, so in that case, uh, uh, first such effort was initiated by Alfonso de Candolle. So where he has carried out that particular, he has understood the importance of conservation of germplasm, and based upon the distribution of that particular wild relatives, their history, linguistic derivative, archaeology, and pattern of variation, he has initially uh, tried to collect and conserve the germplasm, and later he has published one book, that is Origin of Cultivated Plants. So that was the first step for initiated by Alfonso de Candolle. Then later that particular Otto Frankel, so he has given that particular the term genetic resources which is used by the breeder. And from India, one of the professor, that is Professor B.B. Pal, who has played an important role. He was there in that particular expedition under British India and in search of new genes. Why always we have to find out new genes? Because if new genes are available which are playing an important role for improvement of plants, then and then only we can improve that crop plant for concentrate. If resources are not available, then we are helpless, we are not able to improve, and whatever GM crops are there. So as far as Indian government is concerned, only one crop is till today has released for cultivation, that is only BT cotton. Other crop plants, though these are developed, these are utilized in other continents, but in India it is not released. So as far as our agriculture is concerned, we should always prefer that particular genes which are coming from plant origin. And in that case, if we conserve that particular plant, then definitely this will be helpful. So this is the particular Vavilov, Nikolai Ivanovich Vavilov from Russia. He has uh, explained extensively, visited different parts of the world, and he has proposed that particular center of origin concept, where he has proposed 8 centers of origin. So these are this particular center, that is I will show that particular figure afterward. And along with that, he has proposed one law, that is law of homologous series of variation. So whatever character is present in one species, its related character may present in related species. So that particular character we may find in that related species which are coming under that genus, or those genus coming under one family. So if a trait is present in one genus, then its related trait may observe in the other genus from the coming that family. So these are the particular centers proposed by Vavilov, that is China, Hindustan Center, Central Asia, Asia Minor, Meridian Center, Abyssinian Center, then USA, and these particular three centers of South America. Later, he himself modified these 8 center into 11 center by incorporating one center, that is America there, and then Hindustan center was divided into 2 centers, and this particular South American center again divided into two additional 2 centers. So these are the particular crop plants. So where he has explained how that particular center of origin is there, that is primary center and secondary center. So what in by this one, where maximum diversity present in the primary center, and secondary center of origin means where through the migration. So how that particular Gondwana land, human population has migrated to different parts of the world. So along with their migration, they have introduced this plant into different area, and where they have again shown the diversity. So that is known as secondary center of origin. So these are the particular points. Now, in that case, again, he has explained how that particular micro center is playing an important role. So micro center is that one which is showing maximum diversity. Micro center is showing maximum diversity of crop plants. Hello, someone is speaking, please.
Yeah, sir, it is muted, sir. Okay.
Yeah. Now, in that case, if we want to go for germplasm conservation, then how that particular germplasm conservation is carried out? So germplasm conservation is carried out with the help of ex situ and in situ conservation. So in situ conservation is that one where we are trying to conserve that germplasm in its original form, whereas ex situ means away from its original location. So that in situ conservation, where those plants are showing maximum diversity in that area, then that area is declared as a natural park or biosphere reserve or gene sanctuary, where we are protecting that particular area by applying stringent laws, so that that plant material or that wealth of plant will be protected. So in India, such kind of gene bank or gene sanctuaries are established by NBPGR. In Meghalaya is there, the northern states for Musa, Citrus, Oryza, and Saccharum, like that. So what are the merits and demerits? So where without disturbing that particular diversity, we are conserving them, but demerits are there. Moment we declare that this is a protected area, then everyone is anxious. What is there? We should go and visit, and we should take something. So then that is the demerit. And again, natural calamities are there. Fire is there, or your flood situation is there. So that may affect the particular area. Ex situ conservation. So where we are trying to collect seed material or plant material from its original location, and we are conserving them into a different location with the help of seed bank or plant field bank or shoot tip bank or cell organ bank or DNA bank. Now, in that case, what we have to do, we have to, if we have to establish that particular seed bank, then we should visit that particular area, and then we have to collect that particular material. So sources of collection, center of diversity, gene bank, and so on. So priority of collection, in that case, we should find out which plant we need to collect. So that particular priority should be there. That plant is whether coming under a rare category or endangered or critically endangered, and so on. So that particular method of status we should understand, and then we have to go for collection. So where that particular sample, 50 to 100 plants or per sample site, and 50 seeds per plant. So by this way, we have to collect that randomly or selective sampling we have to carry out. Along with that, we have to maintain some field records, so that these field records will be helpful for further characterization of that germplasm. So merits. So where whatever demerits are there, so from in situ conservation, so it is difficult to maintain that one. So that problem is not associated. So we can collect that plant material from all over the country, and we can deposit and conserve them at one location. So that material will will be conserved for years together by keeping some ideal conditions, and whenever this particular material is required, then this particular material is provided to the end user, so that he or she can be used for that material for further improvement program. So demerits are there. So during conservation, some diseases or insects or new weed seeds may come along with the seed material. Then it will be also again showing some problems with the collector. So if that particular collector is giving misidentification, or it is showing some problem with the transportation or lodging, then material will get lost. Then along with this, that particular researcher who is visiting in different area will face different challenges like wild animals, snakes, lions, and so on and so forth. And again, transportation is a huge task as far as collection is concerned. So this ex situ conservation, in that case, first one, that is seed banks. So these seed banks will consist of two types of seeds based upon their particular storage capacity and moisture content, that is orthodox seed and recalcitrant seed as per the Roberts. So orthodox seed, where we can be more, conserved with 5% moisture content is concerned. So whatever cereal, pulses, and so on and so forth, those seeds are there, these are coming under that particular orthodox seed type. And these are the particular photographs which shows that how that particular seed that method of conservation we are even we are following in our home also. We are going for sun drying of these particular seeds, and then we are preserving for further next year. Recalcitrant seeds, so where that particular moisture content can be maintained between 12 to 30%, and that particular seeds especially from fruit crops are there. And these particular seed, if that moisture content goes below or above, then that seed viability will be lost. So where that particular recalcitrant seeds are maintained, especially from the fruit crops. So these are the particular seeds from coffee or cocoa, that is Theobroma cocoa, that is used for making chocolates and so on. And so these are used. Now during seed storage, how we have to store that particular seed? So where we have to maintain temperature, moisture content, and based upon that, duration will be different. So base collection, where we can keep that particular seed for 20 years, then the temperature will be under minus 20 degrees Celsius, and with moisture content for 5 degrees Celsius, 5% is there. So that is called as base collection, and this material is never used for distribution. Only when that active collection seed is showing that particular loss of or germination percentage is less, then and then only this base collection is used. Active collection, so it is stored at 0 degrees Celsius, having moisture content from 5 to 8%, and it can be stored for 10 to 15 years. Whereas working collection, so this particular collection is basically used by the breeders as far as improvement of crop plants are concerned, and these are readily shared among the institutes, among the breeders or NGOs, and so on. So where 3 to 5 years storage capacity is there, or the duration is there, with 5 to 10 degrees Celsius temperature and moisture content between 8 to 10%. So if that particular material, if we have to use, then we should use only from working collection, or we can get material from any of the institute from this one. So these are the particular photographs which will show how that particular material is conserved. So in India, germplasm conservation is carried out on large scale at NBPGR, National Bureau of Plant Genetic Resources, New Delhi, in IRI campus, where entire germplasm of our crop plant is conserved there, and it is 3-storied underground building. Underground 3-story building is there, and no one is allowed to enter. Only with specific precautions, that particular breeder or that particular scientist can go and check that particular status of that particular material. And this is very important as far as conservation and further improvement of crop plants are concerned. Now these are the particular gene banks which are reported for different crop plants in India. So cotton, plantation crop, potato, tobacco, and so on. So these are distributed in a different part of the country. Similarly, that CGIR, Consultative Group of International Agriculture Research, so under their guidelines, different international institutes are also established as far as conservation is concerned. So that IRI is there, IRI for rice, CIMMET for wheat and maize, and so on. So where third world country, where poor crop yield is there, crop management is not there. So in order to improve that particular livelihood of third world country or underdeveloped country or developing countries, so such kind of institutes are established. So such kind of 12 institutes are there which are established in different part of the world, and they are providing not only improved crop plants for the inhabitants, but along with that they are giving best training for scientists as well as farmers, so that these particular techniques
सरजी,
yes
सर पीपीटी स्लाइड शो झालेला नाहीये, स्लाइड शो करा ना त्याला सर प्लीज.
स्लाइड शो केव्हाच केलाय सर मी.
पण ते काय होते, बहुतेक नेटवर्कचा प्रॉब्लेम आहे, फक्त पहिली जी स्क्रीन आहे तीच दिसते, पुढे मूव्ह झालेलं नाही. परत एकदा थोडस स्लाइड शो करा, बाकी काही नाही.
कारण माझ्याकडनं मी तेव्हाच हे केलं.
झालंय का? झालंय का?
हा ते तेव्हाच हे केलं.
मे बी नेटवर्क इश्यू आहे, त्याच्यामुळे थोडस ते दिसत नाहीये. बाकी ठीक आहे, आवाज मात्र व्यवस्थित येतो, काय अडचण नाही.
आता दिसतोय सर? आता स्लाइड दिसतायत का मला सांगा.
नाही सर, पहिलीच दिसते ती.
एक मिनिट मी परत एकदा हे करतो.
ते शेअर परत अनशेअर करून शेअर करा सर.
बर, ओके. नाउ
yes sir, now it is visible.
With the slides are moving?
Yes sir.
Okay. Sorry, तुम्ही अगोदर सांगितलं असं तर मी.
It is a network issue sir, no problem.
Okay, yeah.
Okay sir.
Yeah, these are the particular facilities are available at NBPGR New Delhi, where we are trying to conserve that particular germplasm, and that particular germplasm is distributed. So these are the particular gene banks which are established at a different part of the country, crop by crop, and they are trying to conserve the germplasm. Now slides are visible?
Yeah sir, yeah.
Okay. These are the particular international institutes which are established in different part of the world, especially third world countries or underdeveloped countries, where because of lack of facilities or improved cultivars, the continuous famine is there, or the yield is very less. So these institutes, they are providing the improved plant types, they are giving the knowledge, they are developing the human resource, so that these human resource will go in their respective countries and will help their inhabitants in that particular area. So these are the 12 institutes internationally established in different part of the world. Okay, along with this, again Svalbard. So this particular institute is there, which is present in Norway. So where entire germplasm is conserved in Norway, where throughout year it is having temperature less than -20. So nothing will happen if something goes wrong with these about 12 institutes from all corner of the world. Then this particular germplasm is conserved. So around 7.74 lakh accessions from different crop plants, these are conserved in Svalbard, so that in future, if because of natural calamity or man-made calamities that germplasm get destroyed, then this germplasm can be used for further conservation and improvement. Then plant bank. So this up till now we have learned about the seed bank. Now plant bank, in which we can bring out that particular seedlings from its original location, and then we can conserve them in different locations, just like gardens and so on. So in that we are trying to increase the diversity, but limitations are there. It requires large area, expensive, then damage. It is prone to damages that is caused by insect and pest. Then man-made activities are there, the natural disasters. So these are the particular negative points for that. Shoot tip banks. So in that case, again, we can conserve, say for example, we find a particular elite plant type, and that elite plant type is having a property of nutritional value is there, or resistance to biotic or abiotic stress. So that particular plant material we can conserve by using that particular shoot tip, where we, with the help of tissue culture technique, we can use that particular meristem or axillary meristem and apical meristem, and then we are trying to conserve that particular plant material by using tissue culture technique. So where we can maintain that plant for 6 to 36 months in that particular condition, and it require less space for conservation of that particular plant material. So these are the particular examples how we can conserve that particular plants at laboratory level. Then cell and organ banks. So in that case, we are trying to conserve that particular plant material by using that particular cell or organ. Again, we can conserve them at minus 196 degrees Celsius temperature, and then we can do that particular conservation. So evolutionary conservation and selective conservation, these are the two types, where evolutionary conservation means on its natural way. So these particular plants are evolving, and then we are trying to conserve. Or selective conservation, we are applying some pressure, that is we are putting some pressure in the form of either pathogen or in the form of any stress condition at cell level, and then we are trying to find out any variation is observed in that particular plant. Then DNA banks are there, so where we are extracting the particular DNA, and those sequences are conserved there by using the different vectors, and these can be used further for further studies and so on. So significance of germplasm conservation, if you see, so in that case we can identify, we can find out the abiotic or biotic stress resistant germplasm from that particular collection. So abiotic stress means drought or cold or flood tolerance or salinity tolerance. So if such kind of particular plants, if we identify, then these can be used for further breeding program. Or in case of biotic stress, where fungi, bacteria, viruses, insect or nematodes. So if those plants through screening, if they show particular resistance against these particular pathogen, then these can be used for further breeding program. Similarly, utilization for cropland. So once we know that through this particular collection and their particular screening, these are the particular plant types which are having these properties, then these can be used for breeding program as a parental material. Then somewhere in some crops where that pre-harvest sprouting is there, in case of wheat is there, or in case of mung bean or urd bean is there, so seeds are showing germination while they are present on the plants or while they are present in the pots. So in that case, if you find out that particular plant which is showing that tolerance, then one can use that one as a donor parent and start a breeding program. Or shattering resistance, so there are certain plants which are showing pod shattering is there, that is seeds are splitting, especially in case of soybean is there, or in case of rice, that particular grains are showing shattering from the panicle, and then there will be great loss. So if you find out such kind of resistant plants or tolerant plant which can be used as a breeding material, then plants with highest medicinal value, through screening we can find out that one, and then these can be used. Or if we go with that particular genome sequencing or allele mining or association mapping, so these are the particular further studies after conservation for characterization of germplasm, where we are trying to know what is the particular genome sequence, what is the particular position of gene, then what are the different alternative forms of these particular genes, their position on the particular chromosome. So once we know this one, then with the help of marker assisted breeding, we can start a breeding program, and we can expedite the speed of that particular breeding program, so that we can give that particular product to the farmer or end user. So these are the particular importance of this genome sequencing or allele mining or association mapping. Then conservation of rare plant. Rare means rare, endangered, and threatened. So if you find certain plants which are having some problems as far as their propagations are concerned, then one should conserve these plants. So unless and until we are not finding out that status, then we are not able to initiate such kind of studies. So through collection, we came to know, yes, these are the plants which are rare or endangered or threatened. So there is immediate need for its conservation. So through biotechnological tools, we can conserve them, as well as if some plants are having that particular medicinal value, then through biotechnological tool, we can increase that particular medicinal component so that it can be used for on a commercial scale. So how that particular germplasm is used? So germplasm, so directly when that particular material is not available, as far as resistance is concerned, for concern biotic or abiotic stress, then the germplasms directly can be introduced in that particular area. So such examples are there. So soybean and sunflower has been introduced from USA to India, or ridley wheat, which is showing resistance to rust, or these particular wheat lines were introduced from Sumet to India, or rice lines were introduced from Erie to India. So these are the particular germplasm uses of germplasm. Now, wild relatives of cropland. I will skip this particular part. Otherwise, okay, just I will take two examples. So what will happen if we are not understanding the particular role of these wild relatives of weed relatives? So if you see that particular evolution of rice is concerned, so these are the particular types: indica type, japonica type, or and javanica type. So Oryza sativa is there, so that is from southeast and south and southwest Asia is concerned. Whereas in tropical Africa, so other lineage is there. So somewhere at Gondwana land, one common ancestor was there, and from which that particular today's Oryza sativa has been evolved. So if something goes wrong with that Oryza sativa, then what we have to do? And not a single line from Oryza sativa is playing an important role for improvement. Then we should collect these particular their weedy relatives or wild relatives, that is Rufipogon or Nivara or Spontanea, and then we should cross with Oryza sativa so that we can improve that particular plant type. So similarly, that is from African line. So these are the particular different plant types of Oryza sativa observing in different part of the world. So if you see that particular evolution of wheat is concerned, so that wheat evolution, so how today's hexaploid wheat has been evolved, that is Triticum aestivum with AA, BB, and DD genome. If something goes wrong with this particular Triticum aestivum, then what we have to do, with whom we have to make a cross so that we can obtain that particular solution. So these are the particular probable ancestors of today's cultivated wheat and how they are playing an important role. So this one, that kapli gahu आपण खिरीसाठी वापरतो. So this particular Triticum dicoccum is evolved through a cross of Triticum urartu and Agilops peltoides after doubling of chromosome. Then this particular Triticum dicoccum has been crossed with that particular Agilops tauchi, and through which we are observing that particular today's Triticum asticum, that is hexaploid wheat. So in that case, again, how that particular Norin 10 gene, so a Japanese variety, which is played an important role for development of semi-dwarf wheat varieties, which is developed at CIMET at Mexico. So this particular Norin 10 dwarfling genes, that is RHT-B1 and RHT-D1, both showing their location on 2D chromosome, and these were introduced and developed that particular semi-dwarf line with that particular umber color seed. From red color umber color seeds were developed. So these are the particular, okay, I will not go into detail. So these are the particular gene pool concept. Now what in by gene pool concept? In that case, where we are trying to find out how that particular sorghum bicolor or sorghum vulgare is showing easy crossability and showing production of fertile seeds. So if something goes wrong with sorghum bicolor, then we should first find out with whom we can make cross so that we'll get a fertile seed. If it is not working from GP1, then we should find out that particular gene pool 2 or gene pool 2nd, gene pool 3rd. So related varieties, then related genus, so through which we can obtain that particular desired genes. This is about the okay. In case of saccharum, that is sugarcane. So how today's Saccharum officinarum has developed? through a process of that particular evolution, miscanthus, erianthus, spontaneum, robustum. So through which that particular genome, this genome has been evolved. So if we have to improve that Saccharum officinarum for quality purpose or for resistance to biotic and abiotic stresses, then with whom we have to cross? If we know, can we cross with miscanthus or erianthus or spontaneum, then that particular challenge can be overcome. From sugarcane, so these are the particular how that particular plants are showing relation with whom, and if that solution is not observed in the that plant, that is sorghum or maize, so related plant types we have to see, and then we have to obtain that particular gene, and through process of genetic engineering we can transfer. So it is accepted there, because we are taking that source of gene from plant origin only. This is about Kajan. Okay, I will go somewhat. Cotton. Now, if you see that particular cotton, how old world cotton and new world cotton is there, and through the process of polymerization, how today's tetraploid cotton has been evolved. So in that case, we should know which are the particular ancestors of present day cotton is there, and these particular cotton accessions, if we conserve, then we can involve them as a breeding material or parent, and then we can achieve our target. So these are the particular gene pool concept of gene pool of Gossypium, primary gene pool, secondary gene pool, and tertiary gene pool. So through which we can improve our cotton for conservation. Now I will share our example how we are involved in that conservation of germplasm at Shivaji University Department of Botany. So this is the work carried out in my lab with respect to Vigna. So Vigna, that is our Udid and Moog, Matki, and so on. So we have tried to find out what kind of diversity is present in case of Vigna throughout country, and through which we have tried to find out what are the particular wild relatives and cultivated forms of these Vigna species from entire India, from subgenus Ceratotropis. So such kind of diversity is there. So these are the particular diversity of flowers from that particular Vigna. Then this is the particular seed morphology diversity present in that particular Vigna species from India. Then we have find out the particular diversity between that particular seed sculpture, that is seed surface, with the help of electron microscope, and how that particular pattern variation is there. That itself will tell what kind of diversity is there in that particular seed, not only at morphological level but at SEM studies level also. Such kind of diversity is reported. Now, considering this particular diversity, what we have done, so we have tried to find out, can we use that particular gene pool for improvement of Vigna radiata. So only Vigna sublobata is showing easy crossability with radiata, and it is producing fertile seed. But if you go with that particular secondary gene pool and tertiary gene pool, then we require specific efforts, biotechnological tools events are required, where embryo rescuing is there, somewhere embryo is getting aborted during crossing program. So that can be overcome by removing that embryo and culturing on the medium. So we have tried. Then in case of Vigna mungo, so how Vigna sylvestris is showing easy crossability and development of fertile seeds are there. So that information is generated only after collection of germplasm, and further using that germplasm for breeding program, one can know, yes, with whom we can do that particular crossing program. So these are the particular gene pool which are showing somewhat difficulty as far as development of fertile seeds are concerned. Then next example, that is Schwerizia cirrhata. Kate chirite is also common name. So this particular plant is having very importance as far as health point of view is concerned. It is showing anti-inflammatory property, hypoglycemic, hepatoprotective, antibacterial, and so on, anti-cancer. So this particular plant material is growing only in Himalayan region, foothills of Himalayan region. So this one is that Schwerizia cirrhata. So what we have done, we have tried to find out what kind of diversity is present in Western Ghats of India, and we could collect that particular different plant, that is Schwerizia angustifolia, corymbosa, densifolia, Schwerizia lavi, Schwerizia minor, and so on. And then we have tried, so these are the particular sites of collection, and then we have characterized this particular plant material biochemically as well as molecular study, and then we find out interesting result that Sphaerecia minor means having 6 centimeter height only, but this plant is showing maximum similarity with Sphaerecia cirrhata. So whatever in trade market is concerned, so instead of Sphaerecia cirrhata, many of the times they are giving the wrong material, and if we use that wrong material for medicinal purpose, then it will not give that particular effect as far as treatment is concerned. So we, through that particular molecular tool, we have find out how Sphaerecia cirrhata from our area is having similarity with Sphaerecia cirrhata, not only with that particular DNA studies, but it is also showing similar kind of medicinal properties with respect to different medicinal principal components. So one can use this particular information for further utilization of Sphaerecia minor. So that will also help to minimize the pressure on Sphaerecia cirrhata, because Sphaerecia cirrhata is critically endangered now, and it is playing an important role for various purposes. Similarly, another plant is there, Imbelia ribes, or it is also called as in Marathi Wowding. So this plant is also having, so this is the particular plant type. So this plant is having again different type of property, anti-cancer, neuroprotective, hepatoprotective, and so on and so forth. So this plant is again collected from different part of the Western Ghats, and this imbelin content is there. So this imbelin content is playing an important role, and we have reported that particular plant material imbelin, which is having highest imbelin content from one accession from Shimoga district from Karnataka. So this information is very important as far as utilization of seed is concerned. So if we use seed from this accession, then definitely it will be beneficial for the end users and so on. Then from rice, so in that case we have tried to collect the germplasm from only from Kolhapur district. So where we have identified the land races means these are not improved cultivar, varieties nahi hai, hybrid nahi hai. So these are the particular land races which are conserved by farmer for their own use, and they are showing adaptability to the area. So this particular rice which is conserved by farmers, so which are aromatic one, like basmati. So these are the non-basmati cultivars which are reported there, and they are having a different type of property. So what are those properties? Some land races is showing highest amount of iron content, some are showing highest amount of zinc, magnesium, and so on. So to your surprise, I will share some information. In this area, some of the rice land races are commonly preferred whenever there is a lactating mother is there in their home, or old age patients are there, then this rice is commonly offered. Why? Because it is having highest iron content, so it will help to recover that patient, because of consuming high iron content rice, it will help to increase the blood level, or HB will increase, and ultimately recovery of patient speed will be faster one. So our department now, I'm sharing some information about how our department is also playing important role in conservation of plant wealth. So lead botanical garden is there. So our department is having first lead botanical garden from India with the help of Minister of Minister of Environment and Forest and Climate Change. This particular garden has been sanctioned in 2007, and in this case we are trying to conserve the particular germplasm. So we are bringing such kind of germplasm from especially wild plants, or those plants which are on the verge of extinction, or rare, endangered, threatened, and so on. Then we are raising that particular seedlings, and these particular seedlings we are distributing. So palms and other plants, we are collecting that one, and we are not only planting in our garden, but we are also distributing to the other institutes, NGOs, colleges, so that students will understand what kind of plants are there. Along with this, we are also allowing students to visit our garden. So every week there will be one or other tour will be there from school and colleges, and they are allowed to learn what kind of diversity is there. So such kind of diversity is there. So we are selling plants to forest departments and so on. So so this is the particular attraction of our garden. So this is Victoria amazonica, giant water lily is there. So this particular plant is commonly observed in the Amazon basin. So on this plant, it is not fully grown, but if it goes very luxuriantly, then children having a weight of 30 kg can sit on that particular leaf. Such kind of particular plants we are maintaining in our garden. So what is the use? Someone will ask, what is the use of this particular conservation? So this conservation will help for understanding the diversity. Those who are interested to work on that particular plant, they can visit our garden and they can learn. So as far as diversity is concerned, we are having around 1200 different plant types from angiosperm, gymnosperm, and pteridophytes in our garden. So it is around a 40 acres land where we are maintaining this plant. So this plant wise, such kind of diversity is there. So these are the particular diversity. So I have taken photographs from Yadav sir. So Yadav sir is an eminent taxonomist from our country. So who is doing his work regarding conservation and of identification and conservation of these particular lands. So such kind of diversity is there in our garden. So these are the photographs provided by Yadav sir. Just I'm showing to you. So such kind of plant wealth is there in our garden. So thank you very much for allowing me to share my views on this particular aspect. And I invite the students from Sonpet College to pay a visit to our lead botanical garden so that they will understand what kind of work is carried out in our department. Thank you one and all.
Surely, sir. Thank you very much. We are going to visit your department in this month or last week of this month or first week of April.
Yes, yes.
We are planning in that way, sir.
Yes, sir. Yeah, definitely. Yeah.
Okay. Now it's my duty to thanks one and everyone. First of all, I would like to thanks Dr. N.B. Gaikwad, sir, because this was very new topic for our student, but they have enjoyed it very well as far as UG students are concerned. I'm very thankful to you, sir, for such a knowledgeable speech because it was planned on occasion of National Science Day, but due to busy schedule or because everywhere different kinds of activities are going to be organized, but you in your busy schedule you have given time to us and delivered such a nice lecture. We are very thankful as far as Department of Botany is concerned and college is concerned. Thank you very much, sir.
Thank you, sir.
I am also thankful to respected principal, sir, Dr. V.D. Satpute, sir, of our college, giving permission to organize such a great event here. I am thankful to my colleagues, Dr. S.V. Rankam, sir, Dr. Professor Sandeep Kumar Deorai, sir, Vikas Ragole, sir, and all the colleagues, as well as I am also thankful to all the students who have attended this event very very patiently as far as technological aspects are concerned. Thank you one and all.
Okay, sir.
Sir.
Yeah, sir, I will leave now, sir.
Deorai, sir.
Yes, sir.
No, sir. एक minute. Just wait. Just wait. We have to take photograph. Please start your video. I request all the participants to start their video. That's why we can take screenshot for that. सर्वांनी व्हिडिओ चालू करा जेणेकरून आपण एक स्क्रीनशॉट घेऊ शकतो. व्हिडिओ चालू करा तुमच्या सगळ्यांनी. देवराज सर स्क्रीनशॉट घेतला की मला सांगा सर.
Okay, sir.
Okay, sir.
Okay, sir. Yeah, thank you.
Okay, thank you, sir. Thank you, sir.
Yeah.
पूरा है ना,